So good afternoon, everyone. I'm Judy from Hong Kong. So today is my great pleasure to have a chance to give a talk here. Actually, my lab mainly focuses on the basic studies of EVs in liver cancer. We're trying to understand how EVs contributes to liver cancer metastasis. We also made use of cancer patients' blood samples to validate our basic studies by looking at the expressions of the EV protein that we identified by our basic studies. It is always challenging and also time-consuming to collect EVs from blood samples, especially when we obtain a few samples from the clinicians. Okay, so when I prepared this talk, it is a very good chance for me to review the other studies to know more better or more advanced technologies that help us to do the high throughput detection of the EV protein. Actually, after listening to Andrew's talk, I should have asked him for some slides to include in my talk. Okay. So in a clinical setting, immunohistochemistry or molecular tests performed on tissues is a common practice for the diagnosis of disease. However, tissue biopsies involves invasive procedure, and the location of tissues sometimes has made biopsy impossible. It is also difficult to obtain multiple biopsy samples at different time points. So to overcome these difficulties, liquid biopsies has come into place. Liquid biopsies refers to the procedure in which the biocomponents are obtained from blood or other bodily fluids, such as urine and ascites. These biocomponents, including the circulating tumor cells, cell-free nucleic acids, proteins, and EVs. Analysis of the existence or the relative concentration and expression of these biocomponents provide a range of information about the disease. Liquid biopsies compared to the surgical biopsies are more convenient and cost-effective. They do not require invasive procedure or hospitalization. The procedure is simple that enables timely sampling of biofluids. So this facilitates the detection of disease at an early stage, which allows early interventions to improve the management outcome. Another advantage of liquid biopsies is the ease of repeated acquisitions of the bodily fluids. So this allows monitoring of disease progression or treatment response. So it has been well documented that the EVs possess functional effect. So in the context of cancer, tumor-derived EVs have been shown to support tumor growth and progression and metastasis, including drug resistance, and also mediate immune response, immune escapes by communicating with various cellular components within the tumor microenvironment by delivering their cargoes to the recipient cells. In addition to the functional effect, the cargo of EVs has promising clinical relevance.
EVs have characteristic molecular markers. For example, tetraspanins, ALEX, and TSG101 are commonly expressed in small EVs. They also carry a diverse cargo comprising proteins, RNAs, DNAs, and metabolites. The composition of cargo is regarded as the fingerprint of the releasing cells, therefore providing insightful information about the origin and functions of the releasing cells. From a clinical perspective, it is recognized that EV contents hold significant diagnostic and prognostic value. Several studies have reported a correlation between the levels of EV cargo and the tumor stage, as well as their abilities to differentiate between healthy individuals and cancer patients. I have listed a few examples here. The first study was performed on clear cell renal cell carcinoma patients. The data show that CD103 level in the circulating EV is significantly higher in patients with metastasis than patients without metastasis. In the context of colorectal cancer, the CD147 and CD9 double positive EV level is higher in cancer patients and is reduced after surgery. Another EV protein, ITBL1 is found to be upregulated in cancer patients and is higher in patients with distant metastases compared to patients only have primary tumor. In addition to protein cargo of EV, microRNA carried by EV also have diagnostic value. These two microRNA showed a higher level in patients with metastatic prostate cancer compared with non-recurrent prostate cancer patients.
So although the potential of EV as disease biomarkers has been promising, the quantification and detection of EVs in clinical samples remain challenging. First, the bodily fluids contain heterogeneous populations of EVs. It is virtually impossible to isolate pure subpopulation of EV for subsequent analysis based on the current methodologies. The current isolation techniques of EVs are usually multi-step, time-consuming, and low reproducibility that make them unable to meet the clinical requirement. Obtaining EVs in sufficient quantities with high purity from the limited biofluids is also a challenge. The standardization of isolation, characterization, and quantification methods for EV is a key for clinical translation. So in order to detect EV cargo for diagnostic and prognostic application, the development of simple, efficient, and highly reproducible for high throughput isolation and detection of EV is needed. So the determination of EV concentration and subsequent analysis of its cargo expression requires the preparation of a purified EV sample. Therefore, new technologies with efficient enrichment of EV subpopulation and sensitive cargo detection are needed. Some of the advanced technologies made use of EV-specific surface biomarkers or tumor-specific marker that expressed on the surface of EV for targeted EV capture. For example, tetraspanins such as CD63, CD9, CD81 are commonly expressed on the surface of small EVs, making them ideal targets for EV enrichment. The beauty of the new technologies is that they eliminate the need for prior EV isolation, therefore enabling a more streamlined and high throughput analysis of the clinical samples, even when the sample volume is limited. So to meet the demand of EV-based liquid biopsies, technologies with efficient enrichment of EVs and sensitive cargo molecule detection are critical. Antibodies and aptamers that recognizes consensus EV molecular markers such as CD9, CD63 are commonly used to capture small EVs. Detection of the tumor biomarker on EV will involve the tumor protein targeting antibody, aptamer, or other nanoparticle, which are linked with different forms of reporter biomolecules. For example, the enzyme conjugated with the antibody can convert the substrate into electrochemical signals. The fluorescent signals of the probe that linked with the biomarker targeting agent can be directly detected. Signals of some dye or nanoparticle can be amplified upon the excitation by the light source. In some techniques, the capturing of EV on the substrate, biofilm, or metal film will induce changes that could be the oscillation of free electrons, the shift of wavelength, and conductivity. The amount of EV bound is reflected by the signal intensity. So today I would like to give some examples on the methodology advancement made towards utilizing EVs and liquid biopsies for potential clinical practice. So aptamers are short single-stranded oligonucleotides that have high affinity of a given target. Because of their nucleotide-based structures, aptamers are more stable and more accessible to modifications compared to antibodies that are used for capturing EVs. Without the presence of EVs, the aptamer dye are quenched by different means, either by attachment to the substrate or by another molecule. Upon the presence of EVs, the aptamer dye will bind to the EV protein of interest. Removal of the nucleotides then cleaves the EV-bound aptamer, releasing the free dye, and measurement of the signal intensity of the free dyes reveal the quantity of the target EV proteins. So a study made use of the DNA aptamers that are specific to HER2 and EpCAM, which are highly expressed in cancers, for labeling EV. The labeled EVs were subjected to size separation into small EVs, microvesicles, and apoptotic bodies. And the characteristics of these EV subpopulations were validated in the study. And the fluorescent signals reflects the expressions of HER2 and EpCAM intensity. This assay was applied directly to the serum of breast cancer patients at different stages and healthy controls. And EVs in serums were sorted by size into small EVs, microvesicles, and apoptotic bodies. The concentration of HER2 positive EVs were higher in stage 2 patients than stage 1 patients, and also the healthy controls. The EpCAM positive EVs were higher in patients compared to the healthy controls. In the second figure, it compares the expressions levels of HER2 and EpCAM on individuals, EV of a single cancer patients and a healthy control. The results show that most of the EVs of the cancer patients has higher HER2 and EpCAM expression than the healthy control. So this study provides an example of using DNA-mediated approach for simultaneously sorting EV subpopulation and detecting biomarker. AlphaLyser is an alternative immunoassay of conventional ELISA. The sandwich ELISA involves the use of primary and secondary antibodies against the protein of interest. It involves washing steps to remove the unbound antibodies throughout the protocol. So compared to the conventional ELISA, AlphaLyser performs a more direct analysis of biological samples without the need of the washing steps. Based on this sensitivity, small volume is needed, and the whole procedure is much shorter than ELISA. And AlphaLyser involves two beads, the donor and the acceptor beads, which are conjugated with the antibodies. In the presence of the protein of interest, the binding of the protein with the antibodies will bring the two beads in close proximity. The excitation of the donor beads causes the release of the single oxygen that triggers a cascade of energy transfer to the acceptor beads, leading to light emission. So the light intensity reflects the amount of protein binds with the antibody. So when the donor beads and acceptor beads are conjugated with antibodies against the small EVs molecular markers, the number of small EVs can be determined. When a combination of antibodies against EV molecular marker and tumor biomarker, the number of EV expressing that particular tumor biomarker can be measured. So this study was actually conducted by Takahiro's group, who's an audience here. And their group demonstrated and compared the performance of AlphaLyser with the conventional ELISA in detecting CD147 positive EVs in patients with colorectal cancer. So before testing this system on serum, AlphaLyser made use of beads conjugated with different combinations of antibodies against CD63 and CD9 was tested on the isolated EVs. So from this graph, you can see that AlphaLyser was able to quantify the amount of purified EVs in a dose-dependent manner. It could also detect the presence of EVs in culture medium without the isolation of EV, suggesting that it is a feasible way to quantify EV without the need of EV purification. So to further validate the ability of this assay to detect CD147 that expressed on cancer-derived EV, beads conjugated with antibodies against CD147 and CD9 were used. The signal intensity of ELISA was correlated with the expression of CD147 in EVs obtained from various cancer cell lines detected by immunoblotting. Lastly, the assay was performed directly on serum. The results show that the level of CD147 and CD9 double positive EV was higher in cancer patients compared to healthy individuals, and the level was reduced after surgical resection, implicating the reduced signals is originated from the cancer-derived EVs. And the analysis was also done on EV purified from the serum of the same cohort of patients. So both results are correlated well, suggesting AlphaLyser can be a tool to detect EV directly from the serum. So the next method is the surface-enhanced Raman scattering technology, as Anders mentioned before, which is highly sensitive and specific way that enables the simultaneously target detection. The Raman signals refer to the light that is scattered when a sample is irradiated with a light source. The scattering signal is largely enhanced when the molecules are absorbed on a nanostructured metal surfaces, typically gold and silver. The enhancement can be more than 10 orders of signal magnitude, leading to signal amplification and allowing for the detection of extremely low concentrations of analytes. There are two methodologies adopted in the application of SERS tag. In the label-free detection, samples are directly absorbed to the surface of metallic nanostructures. The fingerprint spectrum of these biomolecules are obtained. For the tag labeling way, antibodies or aptamer targeting specific biomolecules will be conjugated with the reporter molecules that enable targeting detection and analysis. In one of the studies that applied SERS technology in liquid biopsies, the authors made use of a layer of polydopamine that can enhance the binding efficiency of EVs to overlay on the glass slide. The polydopamine layer was embedded with the antibodies against the CD63 and other cancer-related proteins such as MIF1, glypican, EGFR. CD63 antibodies mainly for EV enrichment, and the levels of the tumor biomarker expressing on EVs were analyzed. And the EVs were captured on the glass slide by binding to the antibodies. And then the tag, which consists of the antibodies, reporters, and nanostructures, particles of gold and silver was added. And then the signal was then analyzed. So the study analyzed serum samples of pancreatic cancer patients and samples of healthy controls using tag conjugated with different antibodies against the tumor proteins. The use of MIF1 antibody conjugated tag revealed a significant higher level in cancer patients compared to healthy controls. And the cancer patients were further divided into different tumor stages and also cases with or without metastasis. The results show that MIF1 antibody conjugated tag was able to discriminate between cancer patients with late-stage tumors and those with the early-stage tumors. There is also a significant difference between cases with and without metastasis. And tag conjugated with glypican, antibodies were also applied to the same serum sample of cancer patients and healthy controls. Only glypican antibody could distinguish healthy individuals from the pancreatic cancer, and neither tags could differentiate tumor stage nor the metastatic type of the cancer. And finally, ROC indicates that the MIF1 expressed on EVs has the highest specificity and sensitivity for the diagnosis of pancreatic cancer, suggesting that its potential as a EV biomarker for the pancreatic cancers. And microfluidics is another key technology for diagnosis and prognosis under active development. It is a tool that installed with network of channels and chambers that can precisely control the flow of small volume of fluids. Because of its small size, the process time is short and cost-effective. They are very often coupled with biosensors and other amplification systems that I've mentioned before. With the integration of different technologies, microfluidics can provide a more sensitive and precise method in detecting disease biomarker than conventional assays. In case of EV analysis, this standalone device is able to capture and quantify EV and able to detect multiple biomarkers on the EV. So this microfluidic comprises a cell depletion and a EV isolation modules. In the first modules, the cells are removed from whole blood, and then the cell-free blood is introduced to the second modules, where small EVs are separated from other EV subtypes. Below, there's a figure showing that 97% of the cells were removed, and the morphology and the molecular markers of these isolated small EVs were validated. The system was applied to cancer patients and healthy controls. The concentration and size of the small EVs collected by the system was compared to those collected by ultracentrifugation. Both approaches reviewed an elevated concentration of EVs in the cancer patients compared to the healthy controls. But when the amount of small EVs isolated from the same volume of blood was compared, the yield collected by microfluidics was much, much higher. So this is another microfluidic device that comprises an array of 8 microchannels, chambers that enable EV enrichment, analysis of MMP14 level and enzymatic activity. The signal of FRAP probe is quenched. The proteolytic activity of MMP14 cleaved and the quencher to generate fluorescent signals. Another chamber is able to quantify small EVs and the amount of MMP14 on SEV. This device was applied to plasma samples of breast cancer patients and control individuals. The result reveals the upregulation of both the MMP expression level and also the activity in cancer patients compared to the healthy controls. And the level is much higher in patients with metastasis than patients without metastasis. So these results suggest the feasibility of using these devices in clinical diagnosis by measuring the levels of MMP14 expressed on the small EVs. So to end this talk, I would like to highlight that the translation of EV-based biopsies for clinical diagnosis is just beginning and also requires further consideration. First, we need to have promising biomarkers expressed on the EVs that can clearly differentiate patients and healthy individuals. Second, the standardization needs to be established for all procedures, as mentioned by Ring this morning, such as the handling and storage of samples, EV enrichment, and analysis of biomarkers. And the analytic method used has to be highly sensitive and specific enough to analyze the samples with limited amount and needs to be validated in a large and diversified cohort of clinical samples before really applying them at the clinical settings. So that's all I would like to share today. So thank you for your kind attention.